Yeah, so one thing I was thinking about, which actually kind of relates back to what I mentioned before about what I did in my PhD, which was looking at quorum sensing. So quorum sensing is basically a way that bacteria can communicate with each other and control their behaviours. And so we've got quite interested with CRISPR-Cas systems in terms of how they can control their activity, because we had observed that because you've got these immune systems, it actually turns out that the bacteria often can make mistakes and have a sort of an autoimmunity. So there appears to be some downsides to having CRISPR-Cas, and so we were thinking presumably there are ways that CRISPR-Cas is sort of being reduced in expression and then increased when the cells are maybe more vulnerable to viral attack or plasmid invasion and things like that. So that's where I sort of thought with quorum sensing, this is a way in which bacteria can kind of communicate with each other, and importantly that happens when they get to a high cell density. And what we'd been thinking was and it had been put out there by some other groups, that when populations of bacteria get dense, they're more likely to, a virus, if it infects it, will then spread through that population very quickly. So a bit like, if you take a flight, you're stuck into a small space with lots of people, a virus will spread quite quickly. You can, catch a cold. So it's the same, I think, for bacteria. And so we then thought quorum sensing would maybe allow the bacteria to have a low level of CRISPR immunity when there's less risk of infection, and then when the cell population increases, the quorum sensing signals will tell the bacteria to basically boost their immunity to then try to reduce the spread of viruses through that population. So that's basically what we found. We mutated a gene which the bacteria use to produce these signals, to sort of say that they're in high cell density, and we saw that the CRISPR-Cas systems were actually being repressed, or their expression was down. And so the organism we're working on is a bacterium called Serratia, and it has 3 different CRISPR-Cas systems in there. So it's got like a whole range of immune mechanisms, and all 3 of these were actually controlled by quorum sensing. So when the populations got high, the expression went up, and this actually provided greater defense against these invaders. And we also saw that the ability of the CRISPR-Cas systems to generate new immunity was also being controlled by this signaling. So it's kind of a neat way that bacteria are talking to each other to sort of assess if they're in like a risky situation, and then basically boosting their immune system before the virus actually comes along. So I think that's a pretty neat thing that bacteria are doing. Particularly a lot of people tend to think naively about bacteria as being these individuals that are just growing and dividing, but they are actually often talking to each other, communicating, and then controlling their decisions as a group. So